Friday, especially with Monday. Monday off. In the lecture notes that I posted, what are the numbers referred to?
Slides.
Yeah, the slides in the PowerPoint.
Ooh, okay.
I had people ask me that much. What are those numbers? Oh, they're the PowerPoint slides. Just for my So, any questions? How's the notes part? Go back into it? So I've made mention a couple of times about the fact that bacteria have a very short generation time. It's hard to generalize because different species of bacteria have different generation times, but you know, kind of ballpark average is about 20 minutes. So bacterial cells divide every 20 minutes. How come? Why do they have such short generation times? Well, okay, they're not as complex. That could possibly be a factor. There's a bigger factor, though. Well,
is it because they have non-membrane bound organelles, so they're more prone to mutations and outside dangers?
We did talk about the fact that they are more prone to a mutation, really not because of the lack of organelles, but because of their haploid genome as opposed to a diploid genome. Any other thoughts?
They're small.
They're small. What does being small have to do with having a fast generation time? Dr. Roach will kill you if you don't get this.
We got a high surface area to volume ratio.
High surface area to volume ratio. Remember that as you get bigger, your surface area to volume ratio gets less and less favorable. How come? What happens to the surface area as you increase in size? It increases, but by the what factor? Increases by the square of the size that increase. But what does volume increase by? By the cube of the size that increase. Okay, so you get this disproportion between surface area and volume. So think of volume as being cytoplasm. So what's surface area? Membrane. Okay. And bacteria, like any living cell, have to be able to transport nutrients into the cell across the membrane and to get rid of wastes out of the cell across the membrane. Okay? So if you don't have enough surface area to meet the needs of your metabolism, you're in trouble. Well, bacteria, being very small, have a very favorable surface area to volume ratio. Therefore, they can bring nutrients in pretty fast relative to their volume size and get rid of wastes. And that helps to support a higher rate of metabolism. So that is high metabolic rate, which then contributes to their short generation time. So there are some advantages to being small. What is the limit on small size? Making this too hard. What do you need? What do you need to be a cell? What are the requirements to be a cell? What do you think? What structures do you have to have? What parts do you have to have for your cell? To be a cell, you have to have what? You have to have a membrane. Okay, what else do you have to have? You have to have DNA. It doesn't have to be enclosed in the nucleus, which it isn't in prokaryotic cells, but you've got to have genetic material. Okay? What else do you have to have? Okay, structural. You have to have ribosomes so you can make proteins. Okay? And then you got to have this cytoplasm stuff, which is all the biochemicals and enzymes and stuff, right? So can a cell be smaller than a DNA molecule? Can it be smaller than a ribosome? No. So there is an ultimate small limit to cell size. And that's where it gets interesting with virus, because viruses are much smaller than bacteria. If you look at this slide up here, you've got a comparative virus, bacteria, and then here is a cell, and here's the nucleus of that cell. This is a eukaryotic cell. Are viruses alive? No. They really push the limit of absolute smallness, and one of the reasons they can get away with being so small is that there's things they don't have, and they don't have to have. Do viruses have ribosomes? No, they're pretty much some genetic information enclosed in a protein capsid or capsule. Okay. One of the early things that we're going to do in lab is, once you get your feet wet with the microscope a little bit, you're going to look at some of the basic morphological forms of bacteria. And bacteria occur in three major morphological kind of categories, and they are the bacillus, the spirillum, and the cocci. What shape are cocci? They're pretty round, okay? More or less round. Bacillus are rod-shaped, and spirillum have some curvature to them. And there are some variations on types of spirillum. Some bacteria, we won't deal with any of those, tend to be branched, kind of like fungi almost. But the three major morphological forms that you will see and we'll look at in lab are the cocci, bacillus, and spirillum. Those are such defining characteristics of bacteria that early on, when we started to develop some bacterial taxonomy, that was one of the major criteria for putting bacteria into various groups. And so there is a genus, for example, Bacillus. Okay, those are the rod-shaped bacteria. There are several genus of cocci. There are Streptococcus and Staphylococcus and Micrococcus, for example. How do those differ from one another? They're all round cells. Well, Streptococcus, when they divide only in a single plane. And when the cells divide, they tend to adhere to one another and form either packets of two cells, that's called a diplococci, or chains of cells, this is called Streptococcus. Okay? So Streptococci are characterized by cocci cells, each one of these is an individual bacterium, put together into a linear chain. Sometimes cells will divide in more than one plane, and so we can get some packet and cuboidal arrangements. So you can form a tetrad of cocci. You can see it here. Sometimes you will have a packet of cells. They can have 8 cells in the packet. They can have 16. They can have 32. And again, there are some genera that have been named based upon these particular morphological appearances. So one of the old genera is Diplococcus. Okay? There's a genera called Sarcina. And then sometimes cells will divide in more than 2 planes, forming these irregular clusters of cells. And those irregular clusters of cells are either micrococci or staphylococci. Staphylococci are kind of clusters that look like a bunch of grapes, okay? And there's really no uniformity to the cluster. It can really vary from situation to situation. Micrococci tend to have a fewer number of cells in their cluster, and they tend to be a little bit more regular. So you're going to see some of these morphological forms as we do some of the simple staining in lab. We'll start out by looking at some prepared slides, but then you're going to make your own slides. And one of the first things you're going to do is you're going to determine, okay, what's the morphology of my unknown? Because that's going to be something that's pretty important. And then we'll quickly follow that up with something we're not quite ready to talk about yet. What's the gram reaction? Is it a gram-positive bacterium or is it a gram-negative bacterium? And you can begin to whittle down what your unknown is pretty significantly based just upon those 2 pieces of information. Okay? Well, what we're going to see is that bacterial cells are a little bit more complicated than we tend to give them credit for being. Okay? Normally we think prokaryotes, and we go, oh, they're just these simple cells that really don't have any complexity, any diversity. And that's going to be kind of reinforced when you start looking at these things under the microscope. Because if you've got a bunch of these little cocci, they're just going to look like these little round circles you can barely see. Even the bacillus, which are a little bit bigger. look like this just little kind of sausage-like. And they really don't seem to have any complexity at all. Well, if you start looking at these prokaryotic cells with an electron microscope, then you start to see some more subtlety. And if you then start even staining cells and looking at them with your standard light microscope, certain staining procedures have been developed that allow certain structures in the cell to be differentially stained. And so you can see an endospore, for example, or you can see a flagellum, or you can see a PHB granule, or a glycogen granule. So this particular slide right here is kind of a representation of some of the things you might typically see in some bacterial cells. Most bacterial cells won't have all of these structures. This is kind of a composite of the possibilities. But there are DNA molecules in the cell. They're clustered into a region called the nucleoid. They don't have a nucleus because it doesn't have a membrane. Some bacteria will have flagella. Some will have these little projections sticking out of the cell called fimbriae. Others have structures called pili. Slime layers and glycocalyxes and capsules are cell structures. There's some inclusion bodies. And so, they're a little bit more complicated than we give them credit for. And identification, particularly when identification was first starting to be established, and in some of the older editions of Berge's manual, which is going to be our tool for identification, the schemes for identification rely pretty heavily on some of the structural information. So that's what we're going to spend a lot of time in the lab the first few weeks doing, is learning the staining techniques for the endospore, for the capsule, for granules, for flagellum, etc., etc. And then we can start getting some information fairly early on about what your unknown might possibly be. So what we're going to do is we're going to talk about prokaryotic cells in general and kind of break them down into three regions: the external structures, the cell envelope, and the internal structures. This is a slide that I think that is pretty important because it helps to organize the information. And so if you're not one of the people who like to bring your laptops or something, if you don't print out PowerPoint slides, some of these things that have a little bit more text to them are not a bad idea to print off and have just as part of your notes. So we're going to basically start from the outside of the cell, the external structures, and then work our way inward toward the cell envelope, and then finally the internal structures. And I'll be making some references to things we're going to be doing in lab then to try to allow you to determine, does your unknown have flagellum? Does it have pili? Does it have fimbriae? So we're going to talk about and apply some of the staining techniques that you'll be doing. Okay? So let's start with appendages. So, flagella. We already talked about flagella a little bit in one of the other class periods. And we came to the conclusion that flagella are not unique to prokaryotic cells. Eukaryotic cells have flagella as well. Zach said, sperm cells are flagellar. That's absolutely true. But the structure of flagella in prokaryotes and eukaryotes is very different. Do you remember anything from cell biology about eukaryotic cell structure? Flagellar structure, sorry. I'll give you a hint. It's a projection of the cell membrane. So the flagella is actually surrounded by a piece of cell membrane. And then there's some stuff packed into that extension of cell membrane. Remember what's packed into the membrane that makes the eukaryotic flagella? Some protein structures called microtubules. Do you remember microtubules? Those are those hollow tubular proteins. And they're arranged in a particular fashion. Remember what the arrangement of microtubules is in a eukaryotic flagella? 9 plus 2. Okay, about half right there. Okay? So there are 9 doublets of microtubules, and then there's a couple in the center, and they're hooked by little proteins called dynein arms. Sounding familiar to you at all? And basically what happens is those microtubules slide past one another as the dining arms are phosphorylated and dephosphorylated by ATP. And the dining arms grab onto the microtubules, and when they do that, it puts a stress on them, it causes them to bend. And so the flagella rotate back and forth this way as ATP causes phosphorylation and dephosphorylation of the microtubule proteins. Prokaryotic flagella, totally different. Okay? Instead of doing this, prokaryotic flagella are a single rod of protein that does this. Okay? It rotates like this, kind of like the prop on a motorboat engine, like a propeller, as opposed to waving back and forth like this. And they do not have that 9+2 arrangement. Well, the purpose of flagella is obviously motility. And we'll talk a little bit about how the flagella generates motion in a minute. And why it's an advantage for some species of bacteria to be able to move. One of the things that will be important to you in identification of your unknown is that there are different flagellar arrangements depending upon the species of bacteria that you talk about. Okay? Some bacteria will have a flagella coming out of only one end of the cell, and that particular arrangement is called polar. Sometimes there will be multiple flagella that will surround the perimeter of the cell. That particular arrangement is called paratricious. So the two major categories are flagella coming out of the end of the cell, polar, or coming out of the perimeter of the cell, paratricious perimeter. Those are your two major categories. Now, there are a couple of little variations on type of polar flagella. If you only have a single flagella coming out of one end, that is called monotricious. Some species of bacteria will have a single flagella coming out of both ends. Okay, that's called amphitricious. Amphi means both. And sometimes you'll have kind of a cluster of flagella all emerging from a common point, kind of a tuft of flagella, not around the perimeter, but coming out of one end. That is called lophotricious, L-O-P-H-O. So monotricious, amphitricious, and lophotricious are the three major polar arrangements. And then paratricious, they're all over the surface of the cell. Flagella are really, really small in diameter. They're about 20 nanometers, so we're down to the next kind of order of magnitude in terms of cell measurements. Remember, typical bacterial cells are in the micrometer range. Now we're talking about nanometer size. Okay? They're so small, as a matter of fact, that you really can't see them with a standard light microscope unless you do some special things. So there is a special stain called a flagella stain that you can do, which will allow you to detect the presence of flagella and look at their arrangement. So in this particular case, this would be a paratricious flagellated cell. So how can you make something that is too small to be seen? It's a distraction. Make it bigger, right? The way the flagella stain works is that the chemicals that are in the stain selectively precipitate on the flagella and basically coat it to increase its effective size to the point where you can see it with the standard light microscope. Is that a tricky technique to be able to do to kind of selectively precipitate chemicals on these little tiny hair-like structures? Absolutely. And years ago, we used to battle away with doing flagella stains in lab. And of a class of this size, probably one person would get a flagella stain to work. Everybody else's would just bomb out. And it's just the kind of thing where if you don't do it routinely and if you don't practice it all the time, it's just not reasonable for you to be able to get a decent result, especially one time. So this is a situation where I will provide you, if needed, with some information about your unknown. If you determine by other techniques that your bacterium is motile, then I will give you the information about whether it's paratricious or polar flagella, because that's going to be one of the dichotomies in Bergey's manual that you're going to have to be able to answer, because it takes you one way if you're polar, one way if you're paratricious. You won't really have to know if it's polar, whether it's monotricious or multitricious or ambitricious, but you need that basic information. There's a couple of other instances where I will either give you information about your unknown or verify it. And one of the things that I will do, because it's so important, is I will verify you for your unknown once you collect the data yourself. Your morphology and your gram reaction. Because if you mess up your gram stain initially on your unknown, and you say it's gram positive and it's really gram negative, then you're going to be off in a whole wrong part of the key, and you're just going to be totally lost, and you're never going to do a proper identification. With morphology, some of you will have little tiny bacteria that you'll barely be able to see, and the question will be, is this a cocci or is this a little short rod? Okay. These morphological forms are called coccobacillus because they're kind of in that gray area between being a circle and being a little short rod. So again, I don't want you to misidentify your unknown because you identified it as a cocci and it really is a coccobacillus. So please take advantage of that once you gather that information and turn it in a little sheet of paper for me with your unknown number, your name, and then your morphological information and your gram reaction. Okay. Might as well announce this right now. This is really the only situation other than flagellar arrangement where I will give you information on your unknown or interpret results for you. Okay. There will be instances where you're doing a stain and you'll go, gee, I don't know if this has endosperms or not. Well, we do controls. We'll do positive control and we'll do a negative control. And I will help you with those controls. But you've got to be able to make the judgment call on your unknown. Okay, so if you ask me about your unknown, I'll say, can't answer it. Okay. So just fair warning. Okay, the flagella in a prokaryotic cell consists of pretty much a rigid rod that then extends through the cell membrane and the cell wall. It bends into this hook region, and then it extends out into the environment through this structure called a filament. The motor for the flagella that causes the flagella to rotate like this is a ring of proteins called the basal body. And they are associated with the cell membrane and then also the cell wall. Now, you might wonder why there are two illustrations here of flagellar structure. Well, we're not quite ready to talk about gram-positive and gram-negative cell wall structure yet, but there are fundamental differences in the gram-positive cell wall structure and the gram-negative. Okay? Gram-negative cell walls, as it turns out, in addition to having a cell membrane, have an outer membrane, it's almost like a second membrane, called the LPS layer. And then they've got this little thin layer of stuff called peptidoglycan. So this is gram-negative cell wall structure. Gram-positive cell wall structure, here's your cell membrane, big thick layer of peptidoglycan, but no second membrane, no LPS layer. Okay? So if you look at the structure of a gram-negative bacterium, it's going to have two rings of protein here, one associated with the membrane, one associated with the LPS layer. Here it's got kind of one ring of proteins. And these are different proteins that make up the filament of the flagella. The filament of the flagella is made up of the protein flagellin. This is a hollow structure, and so those flagellar proteins are made on ribosomes in the cytoplasm of the cell, and then they're transported to the membrane and then moved through the hollow cylinder toward the end of the flagella. So the flagella grows by adding to its tip. So it's constantly transporting out these flagellar proteins, and adding them onto the tip of the flagella, so it grows in length in that particular fashion. What powers the flagella? A hydrogen ion gradient is developed across the cell membrane. And to establish that hydrogen ion gradient, ATP is required. And so the rotation of the basal body is coupled to that flow of hydrogen ions through a membrane channel protein. So it is an energy dependent process. So, when might it be an advantage to be able to move via having a flagella?
Move to areas where higher net nutrients.
Okay, so you might be attracted to areas where there are nutrients that are available. What's another possibility for motility being advantageous? What about the opposite side of the coin of what Austin just said? Are there situations where you might want to get away from something that's toxic? Okay. What are those movements called, either toward or away from a stimulus? What's that phenomenon called? Heard the term taxis before? If you're moving in response to a chemical substance, that is a chemotactic response. If you're moving toward the stimulus, it's a positive chemotaxis. If you're moving away from the stimulus, it is a negative chemotaxis. So a lot of bacteria are both positively chemotactic and negative chemotactic. What other stimulus might be advantageous for causing bacteria to move besides chemical gradients? Okay. Light, absolutely. So some microorganisms that are photosynthetic exhibit positive phototaxis. Okay? So if you're an aquatic organism, for example, if you move to shallower depths of water, you're going to get more sunlight. Okay? Well, what determines whether you're moving away from a substance or toward a substance? Well, there are various receptors, and those receptors can be found in the cell membrane. In gram-negative bacteria, many times those receptors are clustered into a region called the periplasmic space. The periplasmic space is this region right here in white, kind of between the outer membrane and the cell membrane. And when those receptors are stimulated, that will affect the direction of flagellar rotation. Chemical receptors are kind of paired to the basal body and determine whether the flagella rotates counterclockwise or clockwise. Counterclockwise flagellar rotation is referred to as being asynchronous. And basically, that's another word for uncoordinated. And when this happens, the cell goes into something that we refer to as a tumble. And it's not really moving in any set direction. It's just kind of, going out there anywhere. When the flagella is moving in a, I think I got that wrong. Let me double check here. I think clockwise is asynchronous, sorry. When the flagella starts to then rotate counterclockwise, then we get coordinated flagellar motion. And we get directional movement. And instead of a tumble, that's called a run. So up here you can see that signals received by the receptors that reflect flagellar rotation. Counterclockwise results in a smooth, linear direction. That gives you a run. And clockwise gives you this uncoordinated movement, this random movement, this tumble. So basically, if you've got some kind of an attractant, a chemoattractant, when you start getting nearer the higher concentration of those molecules, more molecules will bind to receptors, then you're going to be in a run, and you're going to start moving toward that. But if you then start drifting away, you're going to go into a tumble, and you're going to kind of fall away from that particular stimulus until you pick it up again. And so sometimes the movement isn't just straight linear, you kind of zigzag your way along, but net you're moving in this directional motion toward the attract. In the tumble, you're just kind of falling around, and you're not being attracted to it, and very likely you're going to be moving away from it just by random chance. So run and tumbles give you these directional movements. The flagella are remarkably efficient in terms of being able to power cells. One of the things that you can do to compare relative speed of organisms in their motion is represent how fast they move in a given amount of time per body length. A cheetah can run about 25 body lengths per second, which is the fastest mammal. A polar flagellated bacteria can do about 60 body lengths per second. So they're really booking. That's equivalent to a swimmer swimming about 150 miles an hour. And it's equivalent to a swimmer not moving through water, but swimming through tar, because when you're that small, the friction of the water molecules makes your environment much less fluid, and it's much more viscous relative to your body size. And so when you start looking at modal organisms under the microscope, those that are modal can really zip across your field of view really very quickly. And that's one of the ways in which we determine whether bacteria are modal or not, is just direct microscopic observation. We use a technique called the hanging drop technique, and that's why we have those little depression slides that are in your lab drawers, is we need to do a special technique called the hanging drop to really be able to detect motility most effectively. There are some arrangements of flagella that are a little bit bizarre. This particular arrangement, there are flagella at both ends, and they wrap themselves very tightly around the cell itself and kind of wrap around little grooves that are in the surface of the cell. And then when these flagella contract, they cause the cell to move in a corkscrew fashion. These are called paraplasmic flagella. And these little filaments right here are called axial filaments. Paraplasmic flagella are characteristic of a group of bacteria called spirochetes. And there are some nasty pathogens in the genus Spirochetes, including the venereal disease syphilis. The spirochete actually burrows into the tissue, and that helps it to avoid being destroyed by immune responses. Second set of appendages are the pili and fimbri. And these are also external structures like the flagella is, but they have a different function than the flagella. One of the main functions of the pilus is conjugation. Conjugation is a form of genetic exchange between bacteria. And under the right conditions, two bacteria can become joined by a tube. And that tube is called the pilus. And then, particularly if these bacteria have a small extrachromosomal piece of DNA, remember what these are called? These little small circular pieces of DNA? Those are plasmids. A plasmid can be duplicated, and then the plasmid can move across this conjugation tube or pilus into the cell. And this cell now gains some new genetic information because there are genes on this plasmid. And therefore, this particular cell is modified, it's transformed as a result of conjugation. You can't really get the main bacterial chromosome through here, it's too big. The plasmid transfer through this pilus is not uncommon at all. And if this particular plasmid has a gene for antibiotic resistance, for example, then this is a method by which antibiotic resistance can be transferred from one cell to another cell. Okay? And what's really interesting about this is in bacteria, you don't have to be members of the same species to conjugate with one another. So different species can exchange genes via plasmids by this conjugation process. And so we can transfer an antibiotic resistance gene from a staph bacteria, for example, which is a gram-positive, to an E. coli, which is a gram-negative. And so bacteria are pretty promiscuous in terms of exchanging genes, particularly via plasmids. And that really brings up some interesting issues we'll talk about later on. We talk about taxonomy. Because normally the concept of a species is members of different species usually don't exchange genes. Well, because of conjugation now, we've got things that we call species in bacteria, but they are able to exchange genes. So the species concept gets a little bit fuzzy in the bacterial world. There are some roles that pili also play in attachment of cells, particularly to surfaces. Fimbri are shorter than pili and are primarily involved in attachment. Cells attaching to one another and cells attaching to surfaces. So here's a fluorescent antibody stained electron micrograph of bacteria with fimbri protruding from their surfaces. It's almost kind of like Velcro sticking out there. And here you can see fimbrinated E. coli cells attaching to intestinal cells. These are the microvilli of the intestinal cells. And this allows those cells to become kind of stuck to the surface of tissues. And that can be an important factor in pathogenesis. Because one of the things that your body tends to try to do is to flush cells out of the cell by movement of fluids. Well, if those bacteria are stuck onto the surface of cells, then even with this flushing action, they tend to maintain their position and then can invade the tissues and infect them. So the gonorrhea bacteria, for example, has all these attachment structures, pili and fimbri, and they can colonize the urinary tract, for example, even though there is that constant outflow of urine, which would normally wash the cells out of the body. Here are a couple of cells, 3 cells. They are both fimbrinated. It's hard to see, but they've got this kind of fuzzy little hair-like structures out here. But then here are the pili. Sometimes they're called the sex pili. And these are the ones that form the conjugation tubes that allow plasma to be transmitted from one cell to another. Another external structure is a structure called the glycocalyx. And glycocalyx literally kind of means sugar coating. Basically, it is a conglomeration of polysaccharides, some proteins that are excreted out onto the surface of the cell. And the glycocalyx has a number of roles. One of the roles is protection of the cells. One of the things that can help to protect the cell from is desiccation or drying out. Another thing that it can help to protect cells from is your defense process, phagocytosis. And then the glycocalyx also plays a role in adherence. The two most common examples of glycocalyx are the slime layer and the capsule. Slime layers tend to be a little bit less organized, a little bit less dense and compact, and their primary role is to prevent bacteria from becoming too dried out from desiccation. I don't think we will have any instance where we'll see a bacteria with a slime layer. We will, however, be able to see bacteria with capsules, and that's going to be something that's going to be an important characteristic to determine whether your bacterium is encapsulated.
How do you view it to see a slime layer? Like if you
Typically, and I'll show you this with the capsule in just a second, if your cells have these structures, then it has an influence on the appearance of the colonies that make up that cell. And so cells that have slime layers have kind of slimy looking colony structure, kind of wet and glistening and things like that. So you can tell a lot about individual cell characteristics sometimes by looking at the appearance of the colony. Okay, good question. Okay, well, the capsule has two major functions, and one is for adherence. Cells that are encapsulated can stick to one another and stick to surfaces as well. But one of the big things that capsules do is they really are a structure that helps to prevent cells from being effectively phagocytized. As a matter of fact, we'll talk about this later on, but capsules are considered to be anti-phagocytic factors. Okay, and to retard phagocytosis. I'm assuming you all remember what phagocytosis is, right? Where a white blood cell gobbles up and engulfs a foreign invading cell. You may or may not remember, but in Cell, when we talked about the history of DNA, and particularly the question of, what's the genetic material? I introduced the concept of Griffith's experiments with transformation, and we talked about the fact that he was working with these different forms of bacteria, the rough form and the smooth form. Do you remember that at all? Rough forms lacked a capsule and weren't pathogenic. Smooth forms were encapsulated. They had a capsule and were pathogenic. Why were they pathogenic? Because the capsule helped to protect them from phagocytosis. the non-encapsulated forms, the rough forms, they were eaten up by the phagocytic cells. So, here's a rough form of Streptococcus, and here is a smooth form of Streptococcus. These cells are not capsulated. These cells are encapsulated. And again, you can see a difference in the appearance of the colonies. These look smooth in appearance. These look rough in appearance. Okay? There are several techniques you can do to detect the presence of a capsule. This is the most simple one. This just involves suspending a bacterial suspension in a little drop of India ink. India ink, if you're not familiar with it, is just a bunch of colloidal carbon emulsified into solution. And so all these little black dots right here are little colloidal carbon, and it's all kind of bouncing around by Brownian motion. And you can see that the cells here that are capsulated are surrounded by this halo. Well, the colloidal carbon can't come in contact with the cells because of this polysaccharide covering the capsule. There's another more sophisticated stain that we'll use called the Vanderbilt stain, which allows the cytoplasm of the cell to stain one color, the capsule then is a clear halo, and then the slide itself, the background, gets stained another color. So you can see a nice kind of differentiation between the cell and the environment, and then that clear area is the capsule. This shows you the role that the capsule can play in adherence to tissues. And again, these polysaccharide molecules forming this capsule structure can attach to the surface of cells and stick the cells there and prevent them from moving on. One of the things that you will see in a couple of weeks is we will grow bacterial cells in liquid culture in broth, and we'll look for the patterns of growth in that broth. And one of the patterns of growth that's pretty dramatic when you see it is that the bacteria will tend to form a scum that forms kind of a skin at the top of the tube, and this is called a pellicle. If you have a pellicle, then your cells are going to be either fimbrinated or encapsulated, but they're going to be sticking together. They're going to be sticking to the walls of the tube. They're even going to be sticking to the surface tension of water molecules, and that'll form that scummy type structure. Where we'll pick up next time is we'll talk a little bit about biofilms. Because biofilms and the ability to form biofilms are a consequence of whether a cell has some of these sticky structures, fibrils, capsules, etc. And biofilms are something that we're just starting to appreciate the importance of, particularly in disease. So we'll talk a little bit about what biofilms are and the role they play in disease. Okay, so have a great Labor Day weekend. Monday, Tuesday, we will continue on with bacterial structure. I'll probably send you via email over the weekend some recommendations if you have masters for some of the sections you might want to look at that relate to cell structure. I'm also going to put together a little sheet that explains how you can do some optional point activities with mastering if you want to take some online quizzes. And so I'll probably do that this weekend too. And so we'll have class on Tuesday and then first lab on Wednesday. Okay? No course code. No, I'm not running, assignments and stuff. So when it asks if your instructor providing the course code, just say no. You're mainly going to be working out of the study area.
Study area, that's
Yeah.
what I figured.
Yep.
Yes. Something in my future. So when we were talking about the food reg, say you didn't have any prior understanding, or I guess you didn't know what the species was you were looking at. How would you look at this and say that it's just And because like you're talking about the
Fimbri tend. Okay, good question. Good question. Fimbri tend to be shorter.
I don't know.
The flagella,
flagella,
if this were paratricious flagella, the filaments would be much longer.
Okay.
These are a little more kind of stubble. What you got going here is opposed to a beard.
right.
You know? Okay.
So it's a pretty clear difference.
So it's pretty clear difference, yeah.
Okay. Cool.
Yep.
I was just curious. I didn't know because for me, from the blind eye.
Yeah. Yep.
Okay. Cool. Thank you.
Yes, sir.
I'm applying middle school this fall, so I was wondering if you, by any chance, would write a letter of recommendation.
We're going to have to come in and talk because I really haven't had much contact with you, Austin, since your freshman year.
Exactly. So I just
So why don't you make an appointment with Natalie and let's come in and talk about it.
sounds great.
Okay?
Okay, it's just Franklin College. I can get on Facebook or anything else. But I don't get on.
They're having real problems with their website right now.
Yeah, I can only get on the division of science.
I tried to get on yesterday, but it didn't work. But I was just on it just now. It worked.
It just, natural science
Yeah.
just worked out?
It worked for like
On the
everyone
lecture
else in this class, and my computer was like stuck like this the whole time.
Oh.
But it let me get on any other side I wanted.
I got some yesterday in the library.
What were you trying to get PowerPoints or were you trying to get?
Yeah, and the lecture notes.
You know what I might need to do is just send out a class email with lecture notes and
Oh, with
PowerPoints.
like the documents.
Well, I just went on, when my internet worked, I just went on and downloaded all the PowerPoints on my computer. But
Yeah.
then like the lecture notes, it's like a website. It's not like a file, right?
Well, it's just linked to Word documents.
Oh, okay.
Well, no, that's not true. Some of them are actual web pages.
That's what I think, yeah. Because I was like, I tried to download a couple of those and I saw that, so I
Maybe, I mean, since we're having problems with that, maybe I'll just get in the habit of sending you guys, when we get to a new chapter, an email with the files for PowerPoint.
That would be good. Because then we would always have access.
Yeah. In fact, I'll go back and do that right now.
Thank you.
Did you receive Austin's email about?
I did, yes.
Okay.
Did you happen to ask people or get any sense of what day of the week might be better for Dr. Williams?
Oh, no, not yet. We were going to wait until next Tuesday's MASH to make sure we have everyone on their email list.
Oh,
And then-
okay, next Tuesday you're having a meeting? Okay.
We're having the welcome bash.
Okay.
So that's when we're going to put out the email list again, just to see if anyone else last minute wants to sign up, and we're going to be talking and stuff. So.
Okay, maybe I'll email him and ask him if he has a preference.
Yeah,
because,
because Austin, I don't think really
I mean,
matters as long as it doesn't conflict with like anything going on around the school, you know what I mean?
yeah,
So,
okay.
Yeah, go to bed.
okay.